everybody. Good afternoon. Thank you all for coming in for the Wednesday Science Hall Seminar Series. I have the pleasure of introducing our speaker for today. This is Professor Arnold Matthijssen, who comes to us from the University of Pennsylvania. He's in the Physics and Astronomy department, but he doesn't only do physics. Professor Matthijssen came to us originally from the Netherlands. He did his undergraduate work at University College in London, England, and then followed that up with a PhD from Oxford University. It's kind of prestigious. He did some postdoc work afterwards at Stanford University, which is like American prestigious. And now he's running his own research group there at UPenn, and he studies basically the physics of microbiology using fluid mechanics. So I'm going to give it away to Arnold, and look forward to it.
Thank you very much. All right, then. So what I want to talk about today is about collective functionality that we see in biology. And I've got a picture up here, which is perhaps familiar to most of you. It's a school of fish, right? So the idea is that these fish are having shared functions, that they can do things together that individually they cannot. In this case, maybe they are protecting themselves from predators, a shark that wants to grab a single fish, but because there are so many of them, the shark gets confused and then they can protect themselves. And this really happens, everywhere in biology. So it happens, in these schools of fish, but also flocks of birds, and maybe these herds of sheep that are not just able to stay warm together as a community, but they can even do collective navigation. You can see that they're going through a little gate over there. And so the sheep back there in the right bottom corner, they don't really know about that gate, but they just follow all the other ones. And collectively, they can achieve a function that individually they cannot. Now, this really happens not just at large length scales, but everywhere in biology, also at much smaller scales. Here, this bottom left picture is all about cilia, the airways. So these are small beating organelles that are transporting all the bacteria and all the viruses that we breathe in every day out of our lungs. A single breath contains like 100 bacteria and about 100 viruses, and they all get, cleared out of our lungs by the collective transport mechanism from these cilia. It's like a little conveyor belt, if you wish, where the particles get transported out of our lungs. Also bacteria, they can push onto each other, and that's how they can spread out over surfaces, breaking the forces of surface tension. And even inside cells, our cytoskeletons are made out of these microtubules that have collective interactions that keep the integrity of the cell in place. So that's the main idea, that there are shared functionalities in biology. And as a physicist, that's what I'm trying to understand, to see if there are microscopic interactions that lead to these large macroscopic phenomena that we can then try to understand. And so here in this picture, you can see how that kind of builds up in terms of length scales. So it starts all the way down here at the nanoscale, like really single molecules, like, I don't know, molecular motors or enzymes. And together, they have shared functionalities. Together, they give rise to cell dynamics. Now, these cells, they start cooperating together. And if my button works, then you will see. Okay, great. So these cells will work together and they form tissues. And these tissues then again will start working together. And now my screen will die completely. And that leads to organs and organisms and even groups of organisms that will then work together over, now orders of magnitude in terms of length scales and also time scales. So that's the general picture. Can we try to understand, as physicists, the basic rules of interaction that leads to these emergent phenomena, that leads to self-organization across these much, much larger length scales? The first thing that I would like to talk to you about today is about collective functionality in terms of communication. The idea is that cells can send messages to each other. And maybe let's think about communication between cells in our own bodies. On the one hand, there are signaling molecules, things like adrenaline. And if, someone will tell you a horrible joke, then they might get excited because of some adrenaline that pumps up in your body. And there are some advantages and disadvantages to that. On the one hand, adrenaline is like a global signal, right? So it reaches every single cell in your body and it therefore is very robust. On the other hand, it takes a long time for these signaling molecules to dissipate. And they're also not very specific. Because they reach every single cell in the body, you can't really target a specific area. Now, on the other hand, we have neurons, which are much more specific, right? They're electrical cables, they run everything in our body, and they're super fast. So you can send a signal, you know, within milliseconds from your brain to your muscles, and that's why I'm able to wave my hands around at you guys. And so, you know, those are great advantages, but on the other hand, that makes them also much more sensitive to damage and prone to specific diseases as well that target this specificity. Is the contrast of this screen good enough for you guys in the back? You can just about see it? Okay, good. So on the one hand, we have here these adrenaline or signaling molecule types of communication, and they are hydrodynamic. They often go through the cardiovascular system. And on the other hand, we have neurons, which is more like a trigger wave, where you have one cell that will trigger the next one and the next one. And so there are waves that will propagate throughout our body. What I would like to talk about today is something in the middle, something that we call a hydrodynamic trigger wave. So we study these single cells in the lab. They're just unicellular microorganisms, and they're called Spirostomum ambiguum. Thank you so much. That's really nice of you. So these cells, they're very special for a couple of different reasons. On the one hand, they're rather big. So you'll see a scale bar up here that's about, you know, a millimeter. So for a single cell, that's a big cell. They're not just big, they're also really fast. So what happens is that when these cells receive a signal, they will contract their bodies down. So they will contract down to a ball like that. Now, this video is slowed down like 100 times. So this whole thing happens in about 5 milliseconds. The blink of an eye is like 100 milliseconds. So this is really, really very fast. And as a result, the forces that are exerted by this cell are just phenomenal. It's like 15 G forces, 15 times gravity. Like a fighter pilot, you know, will faint, you know, around 10 G forces. So that really raises a question of, you know, how can a cell survive these large type of accelerations? And you know, why would you do it in the first place? Well, the answer is that when this cell gets attacked by predators, it will release toxins. So there are these very small vesicles that are attached to the cell membrane. And as it contracts through this liquid, it will spread out these small toxin vesicles, and that way it can paralyze predators, and that way the cell can protect itself. Now, I've got a slide about the mechanism of how that works. Here you can see that contraction, 5 milliseconds, and then it's totally reversible as well, right? So they'll come back to the original length. That takes a little bit longer, maybe like half a second or something, and they can just do it again and again. The way that it works in terms of biochemistry is, on the one hand, mechanosensitive ion channels. So these are channels that will let calcium molecules through. Now, you stretch these ion channel, the calcium will flow in, binds to these long filaments, these long proteins that are called myonemes. And on that binding, you get a very rapid contraction, like basically the molecule collapses onto itself. And that leads to these really strong contraction forces. And then slowly with time, there are calcium pumps, and they will slowly pump out the calcium. And then they will unbind from these myonemes, and then the cell will slowly relax again to its original length. Now, we did some measurements of this to measure, you know, the velocity of how fast these cells are contracting. And what we see is like spikes in the velocity, you know, on the order of, you know, maybe 0.2 meters per second. So we're talking about human walking speed type velocities here. And again, accelerations, like I said, of about 15 G forces. So really strong accelerations in these systems. Now, it's not just that these cells will protect themselves individually, but they can do this together. And they can do that by communicating with each other. So in the next slide, you'll see the same cell that's now sitting in a liquid. And the liquid, is seeded with these small tracer particles. And we look at this with a high-speed camera. So when I play this video, then you'll see that as the cell contracts, it will generate these vortex flows. Now, the Reynolds number of this liquid is much, much smaller than 1 when the cell swims around, which means that viscosity really dominates the fluid mechanics when the cell is normally moving. But then during that contraction, the accelerations are so large that that Reynolds number shoots up to like over 100, and therefore you now go from the laminar flow regime to the turbulent flow regime. So you literally see vortex shedding. There is this like a small instance of turbulence, and that helps with spreading out these toxins into the environment. On the other hand, it's not just the spreading of the toxins, it's also a signal that is spreading out. Because if there is now a neighboring cell, it will feel those flows and it can respond to it. So we know something about how strong the flows are that are being generated by this cell. And the next type of experiment that we wanted to do is whether we can tell something about receiving these signals as well. So to do that, we build a small microfluidic device, which is basically just 2 surfaces, and one of them has a hole in it. And so you suck out liquid through that little hole, and then you get a flow that goes inwards towards the center. And we call this the black hole machine, because it's a little bit like falling into, a black hole. And so, what happens is that the velocity of the flow gets larger and larger as you get closer to the center, and therefore you are stretching these cells. The velocity at the front of the cell is larger than the velocity of the back, and so that gives you an extensile force. In cosmology, there is a word for this. If you fall into a black hole, there is a process that is called spaghettification. And so you can imagine that there are very strong tensile forces that are now acting on these objects falling into the black hole. And the same thing is happening here, right? So what we do is we can calculate the membrane tension that is exerted on the cell membrane as you get closer and closer to the black hole. That is, the fluid is now exerting stresses onto the cell membrane because of these stretching forces. And the colors will indicate the strength of these of this membrane tension. And you'll see that here, there is a region with like the largest tension, and that's where these ion channels are now going to be pulled open, and then the calcium will flow in, and that will then bind to the myonemes and sets off this cascade inside the cell that leads to these contractions. So the idea is that in this experiment, if we look at this with a high-speed camera, we can tell exactly at what location the cell is going to contract, and from that we can back calculate the tension on the cell. So we can say something about the forces on a single molecule using these microfluidic experiments. And here are the results. So here we go. This is a typical experiment. The cells will come in from all the possible directions, maybe 100 cells in this one experiment. And the colors are an indication of the membrane tension at the moment that these cells contracted. So from that, you can, plot a histogram. And then you have on your horizontal axis the membrane tension. Here, none of the cells contracted. You increase the tension, more cells contracted. You increase the tension even more, and eventually all of these cells will have contracted. So that basically determines the sensitivity of these cells to the signals that they will be receiving. A weak signal, nothing happens. A strong signal, the cell will respond. Now, you can also say something about whether a cell is good at responding to these signals or not. So we can measure the length of these cells and measure their sensitivity. And what we see is that the longer cells are more sensitive. And that kind of makes sense, right? Because if you spread out your arms, then you've got a big antenna, and that makes you more receptive to these signals as opposed to the smaller organisms, which are not so sensitive to the same signals. So here is the organism length, and the big cells, they only need a very small signal, and the shorter cells up there, they need a larger signal. So we know something about the generation of the signals. We know something about the sensing. Now we can try to bring these two things together. Now here is a video of these organisms that typically live together in communities. And what now happens if one of these cells gets triggered here in the bottom right corner, then the first cell will contract. It generates a signal, then its neighbor also gets triggered, and the next one, and the next one. And we see these waves that propagate through these communities. And again, this video is slowed down about 300 times. So the time here at the top is 80 milliseconds or so now. So the velocities at which these signals will propagate through these communities are like thousands of times faster than their swimming speed. So it's a warning signal. A predator is coming in from one side, and it's not just the one cell that's releasing its toxins, but it's all of these cells that are synchronizing the release of their toxins at the same time. And so now a single cell can protect itself against multicellular organisms. It's usually fish and shrimps and much larger types of organisms that will target these unicellular guys. And so this is a way for them to protect each other. You see that, there is this network that forms, this connectivity network, and we can tell exactly whether the cells will be able to sense each other or not based on their relative distances and their relative orientations. So from these experiments, we can build up a theory, and that theory we call percolation theory. And so maybe some of you have seen this type of coffee maker before. The way that it works is, you put some water here in the bottom reservoir, and you put some coffee grounds in the middle, and then you boil it, and the water goes through, and you get coffee at the top, right? Now there are three possible scenarios that you can get. On the one hand, if you put too many coffee grounds, then the water can't go through anymore. And so you end up with nothing. On the other hand, if you don't put enough coffee grounds, then the water will definitely go through, but then you don't have water. Then you don't get any coffee. Then it will just be like, a really weak, kind of petrol station coffee, which is, pretty horrible. And so there is this perfect point in the middle where you just put just the right amount of coffee grounds, and you get like a really nice spread. Are there any Italians in the audience? Yes. So in my experience, Italians somehow genetically have this feeling for exactly how many coffee grounds to put in just to get, the right coffee. So, this is called percolation theory, right? So what that means is that you put just a little bit more coffee, and then suddenly the flows don't go through anymore. So it's very, very sensitive. Very small changes in the concentration will lead to a dramatic effect. And that's what you see over here in this graph. Oh, oops, wrong one. Okay, go back. Okay, great. So look at the blue line here. On the one hand, we've got the density here of the coffee grounds or of the organisms, as you wish. Now, very small change will give you a very big jump. What that now means is that these cells are trying to communicate with each other. If they are too far away from each other, then the distance between the neighbors is too much, and then they just cannot send a signal to each other anymore. So you get zero percolation, you get like no connectivity. Then you go across this one line here, and then suddenly this whole community is connected with each other. However, if you go to really high density, then you basically run out of food, you run out of oxygen, you run out of nutrients. So a biological system does not want to live on the left because you can't communicate, and you also don't want to sit on the right-hand side of this plot because it gets way too cramped. So there is a natural tendency for these biological organisms to live close to criticality. They want to be close to this critical transition because that's where they can tune their communities in the most effective way. So yeah, now we are exploring not just how these cells can communicate with each other, but also how they can process information. And so that's the next part of what I'm working on. Next, I want to ask you guys a question. So it's related to this picture over here. I imagine that we have like a bunch of bacteria that are all swimming in the same direction. So say some cells that are swimming here from the left to the right, and then there is this little particles, in the middle. And the question is whether that particle is going to be pushed forwards by the cell body of these bacteria, or whether it's going to be pushed backwards by the flagella of the bacteria. So imagine, there is this little green guy just like roaming around in the fluid, and it's going to be maybe pushed one way or another. So what do you guys think? Raise of hands, who thinks that this particle is going forwards along with the bacteria? No. Who thinks they're going backwards? Okay, good raise of hands. And who think that they're not going anywhere on average? Okay, good thinking. So let's have a look at this. Here is an experiment where we look at very small microswimmers. This is a little oil droplets that's swimming by itself. So this is a little capsule, if you wish, and you can put some drugs inside and it will start swimming. Now, in the liquid, again, we put a lot of these little tracer particles, and we look at the motion of these tracer particles and how they are transported around by these microswimmers. And what we see is that if there is a particle right in front of it, then it gets pushed forwards. But if they're on the side, they get pushed backwards. So it's this plot over here. Like if they started along this line, and the droplet was moving here from left to right, and all of these guys are moving forwards, and all of these guys, they ended up somewhere at the back. So not really definitive answer, like might be going forwards, might be going backwards. So in a way, what we want is the average of all of these guys. Here is another way of looking at that. Here is our droplet or our bacterium swimming from the left to the right. And then all of these little grid cells that you see here are basically parcels of fluid. This is the liquid that gets deformed because this guy is swimming and it's generating some flows because of its swimming motion. And so that liquid gets deformed. And indeed, again, you see that the particles who started very close to the front will get pushed forwards, and the ones that started more on the side get pushed backwards. Now, there is this volume that we call like the wake volume or the entrainment volume, which is, like how much of that liquid moves along with this swimmer. That volume has a name. It's called the Darwin drift volume. And it's named after Charles Darwin, who was the grandson of Charles Darwin. So the grandfather was the biologist, and the grandson was a mathematician studying fluid mechanics. The grandson was called Sir Charles Darwin, and the grandfather, for some reason, never got that title. So I don't know why. So Darwin drift volume will tell you the amount of liquid that gets transported along with a particle that is moving through a fluid. And what we want to do is to calculate this drift volume for a bacterium or for some kind of microorganism that is swimming around and transporting things. So we can do that. And, we can look at not just a single microswimmer, but we can look at maybe, a whole school of them. Like think of these fish, or think of flock that are all, moving in the same direction. And there you can see all of these little tracer particles that are now moving along because of this collective motion. So again, this is an example of a shared functionality. Like a single swimmer would not be able to transport these guys around, but together as a group, if they're all moving in the same direction, then they can transport all of these particles. Now, it seems like they're maybe not moving all that fast, right? The particles are going much slower than the swimmers themselves. But the thing is that literally anything that's dissolved in this liquid will move. So the whole fluid moves around. So the total volume of liquid that gets displaced is actually 20 times larger than the volume of these swimmers. So you can calculate this drift volume, and you can write down an equation for the fluid mechanics, and you can find out precisely how much that Darwin drift volume is, and it turns out to be like 20 times larger than a single particle. So imagine that you're a bacterium and you're, exploring a new environment. Maybe you're moving into a network, like a porous medium or a flow network or something. Then initially there is no oxygen inside there. But then together as a group of bacteria, you move into this new habitat, and then together you can bring all of these nutrients inside. And it's quite a strong effect. So would be interesting if you want to establish a new niche somewhere. Now, we also thought a little bit about this in terms of, drug delivery, because you could think of these, as micro robots that are maybe transporting some drugs inside, that are actively sitting within these swimmers, but it could also just be passive, particles that now get transported along because of this active motion. So at least from the theory point of view, there might be something interesting there. I'll talk a little bit more about drug delivery in a second, but at least it's an idea. So, active transport happens everywhere in biology. Here is another example. Here are some, a plant, that sits in your aquarium maybe. And this plant is covered in these very small microorganisms. If you zoom in, you see that there are all these small green or kind of bluish organisms. And what they do is that they are all trying to capture food. They're all trying to stir the liquid around. They, again, have cilia, just like the cilia in our lungs. And they generate flows. And every single one of these cells will be propelling the liquid around and stirring this fluid around. So we give this a name. We call it an active carpet, because it's literally, a surface that is covered by these microorganisms that are all pumping fluid up and down. So now you might be interested in, the amount of oxygen or the amount of nutrients that you could bring towards these active carpets because of this active mixing. So that's the problem statement that we have. Here we go. Excellent. So we have a source of nutrients, say some distance away, and then we have a sink. And, that's where the organisms are sitting and they're trying to eat. Now, what these organisms could do is they could just wait for diffusion to happen, right? So, there will be some thermal fluctuations, and then these nutrients will just diffuse around, and eventually they will reach the organisms, and then you can eat them. Or you can be a bit more proactive and you can stir the liquid around, and then you can try to create some fluctuations, and those will then enhance your diffusion and bring in more nutrients. So the questions that we then are interested in is, like, how much food can you bring in this way? So what is the strength of these fluctuations that you can generate? Then what's the diffusivity of these nutrients as a function of the distance from this active carpet? And then, like the time required to bring in a particle, or in the end, the flux of nutrients that you can bring in. So it's a mathematical problem that is basically statistical mechanics out of equilibrium, because you are driving the system out of equilibrium all the time, right? Like all of these organisms are injecting some energy at a small length scale, and they are stirring up this liquid at much larger length scales. And then what they get in return is a nutrient flux. The spoiler is that you can get way more energy from the nutrients than the energy that you put in stirring the fluid around. So this is a very efficient way of bringing in things. So let's try to calculate that. You know, here is a picture that shows you the flows that are generated by a single microorganism. Let's say it's a flow that you kind of pump downwards towards your surface, but then anything that you pull down has to go back up again because there is a surface and you can't bring liquid through the surface, right? So there is a net flow that is averaged to zero in that, you know, vertical direction. And then you just put thousands of those on the surface. Oh, I shouldn't have walked in this direction. Okay, here we go. So, you know, like imagine all of these microorganisms that are now sitting on that surface and they are all pushing and pulling on the fluid and they're all pumping, you know, this liquid up and down. And we want to do some statistical mechanics with this. So the first thing you can do is you can calculate the total flow that's being generated by all of these guys together. And that total flow is that blue arrow up there. And you see that this blue arrow is going to be fluctuating, it's going up and down, and on average is equal to zero. So again, it's a surface, and so you cannot have a flow going through the surface. So the mean that you get is nothing. But you do get some variance. And that variance is going to lead to diffusion. The, you know, distribution of these flows looks a little bit like a Gaussian, but actually there are these long tails. So that means that the likelihood of having a rare event is actually not all that rare. So you get these outliers, which are very good at bringing in a lot of particles. And so there is a strong notion that that not being like normally distributed, not being Gaussian, it will really enhance your transport. And we can calculate that. So, you know, the first thing that we do is we calculate the correlation functions. So there is a memory time scale in these systems. The organisms will take some time, you know, switching between pumping fluids up or down. And so we can map that onto an Ornstein-Uhlenbeck process. So that has a well-known you know, correlation function that has an exponential decay time, and we can calculate that analytically. And then you're in business, because if you know the correlation function, you can integrate it. And that gives you the mean square displacement. And you know, you do two integrals, and if you know the mean square displacement, then you know the diffusion coefficient. Now, what you see is that this diffusion depends on the distance. So if you are very far away from this active carpet, you don't get much diffusion. And if you get closer and closer and closer, then you get more and more and more diffusion. So there is a scaling law in these systems. So it turns out that this is like a 1/z squared scaling. So the further you go away, you get a 1 quadratic decay. So here, if you are, you know, far away, not very much happens. If you get a little bit closer, more diffusion. And if you go very close to the active carpet, lots and lots of diffusion. So what we want to do is to solve the total amount of transport, the flux that you can get from this type of space-dependent diffusion now. So to do that, we can try to write down a Fokker-Planck equation, or Fick's laws, if you wish. Now, the standard laws of diffusion are, you know, something you would find in a textbook. There is a flux that we call J, and that flux is proportional to two things, the diffusion constant and gradients in a concentration, right? Now, the second equation over here is just a continuity equation. And if you put those two things together, you get the standard diffusion equation. But now this D here is a constant. In our system, D is not a constant. It depends on space. And so the question is, should it sit outside this derivative? Should it go inside the derivative? Like maybe between, you know, the two derivatives, if it's a quadratic derivative. So there is some, let's say, uncertainty in the system on how to define exactly what is the valid diffusion equation. It turns out that you can still derive this. You can use kinetic theory, and from that, you'll find two effects. Basically, there is a space-dependent diffusion that comes from two different contributions. On the one hand, you have variations in the particle speed or some kind of effective temperature. And on the other hand, there is a collision rate or a, what do you call it, a mean-free path that could depend on the distance as well. So think of two boxes. One box has like a temperature gradient inside. One side is very hot and you get more diffusion. The other side is cold, you get less diffusion. The other thing is about the mean-free path. You could think of another box that has some obstacles inside. And now these molecules are bouncing up and down and they're bouncing between the obstacles. If you change the density of these obstacles, then you get longer mean-free paths if there are less obstacles, which means more diffusion. And you get less diffusion on the other side where the particles are more cramped. So you put those two terms together, and then what you end up with is a new flux that's called a generalized flux, which is the same thing as this J over here, except now instead of one term, you get two contributions. One of them from the space-dependent temperature, and one of them from the space-dependent collision rates. This is not really temperature, right? This is just the activity of the organisms that acts like a temperature. So two terms in this equation, and we can solve it analytically. And then we can really do some nice calculations. So here's our problem. We have the source, we have the sink. If we solve this problem for standard, thermal diffusion, we know the solution. It's the Boltzmann equation. So you can just write down the Boltzmann solution, and what you get is a density profile that's perfectly linear. That rho is linear in the coordinate z. And the flux is a constant, and that constant just depends on the diffusion coefficient. But now it's not thermal diffusion anymore. We get these living organisms. So we have space-dependent diffusion. So we get this generalized flux, and we want to solve the fluxes for that generalized situation. And we find some very interesting results. On the one hand, we find some kind of self-cleaning effect. So the idea that close to the active carpet, there is less accumulation of particles. Like you can imagine all of these fluctuations that are pushing nutrients away, if you wish. And so just like inside our lungs, there is a cleaning effect where you have lower concentrations of particles close to the active regions, and you have more particles that are further away in the less active regions. The second thing was my spoiler. I said that there is a flux that is now going to scale with the amount of force that you put in, and it turns out that that's a quadratic relationship. So you do 2 times more effort, you get 4 times more food. You do 4 times more effort, you get 16 times more food. So this is a very nice way for organisms to just, pump the fluid around a little bit, and, they definitely get some what shall we call it? What's the expression? Bang for your buck. Oh. That's the one. Yes. So this is just the first step of trying to solve, active transport processes in biology. It's a non-equilibrium problem, but it's a rare example where you can find exact solutions for a non-equilibrium equation. And so now we're trying to think of, maybe generalizations of having like, spherical active carpets, like a cell that is trying to recruit some nutrients from its neighborhood. Or maybe it's something inside the cell, an organelle that is trying to move proteins around. So there's a lot of fun biology that then comes in again. I promised that I would talk a little bit about drug delivery. But there is this one point that I want to talk about first, which is biofilms. Biofilms are basically active carpets, right? They are bacteria that live on the surface. And so they also run out of food all the time because these bacteria are so densely concentrated. So you can now again imagine that little tracer particle, that little nutrient particle, that is going to be eaten by those bacteria in the biofilm. Now here is a video of a biofilm. Here are all these bacteria that are sitting on the surface. They're moving around a little bit. and they're pulling these nutrients down towards the biofilm. So it's an explicit, example of visualizing how many nutrients you can bring close to an active carpet by just doing a little bit of effort. So yeah, you can even calculate precisely the strength of these flows. So you see that there is this blue region where the nutrients are being pulled down, and there is the red region where the liquids are pushed up again. And so this recirculates, and you can calculate precisely the strength of these flows. It scales, with like the density of the biofilm, that's little n here, then the size of the cells, and then kappa is the force, the amount of energy that the cells are putting in order to generate these transports. Now, this is like the perfect, ideal situation, right? Like a mathematician wants to have a perfect, circular biofilm. But in reality, a biofilm is not circular. They look more like this, right? And it turns out that this mechanism is totally robust. So it doesn't matter the shape. Here is a simulation where, the bacteria are now sitting along these branches of the biofilm. And the blue colors are still corresponding to regions where the nutrients are being pulled down. And the red colors is the regions where the liquid is being pushed back up again. So again, you get this attraction of nutrients even for these more complicated shapes. So yeah, it does not really depend on the shape at all. Okay. Yeah, so these active carpets, they're literally everywhere in biology, right? There are bacterial biofilms, the cilia that we find in our lungs. Also, coral reefs are generating flows all the time, not just to bring nutrients in, but also for fertility. They have the gonads that get transported around that way. But also, inside our cells, the cytoskeleton is an active carpet. Cell membranes are active carpets. And so, yeah, we think that maybe these non-equilibrium transport processes might be interesting for some of the processes that are happening inside cells as well. Okay, now the last thing I wanted to talk about is about, again, active transport, but now in a more biomedical setting. And so this is a field that I'm new to, and I'm just starting up my group, but one of my new incoming PhD students had a background in nanotechnology, and so this is something that he really wanted to work on. And so, this is my debut to this field, if you wish. So this is the idea. There's a cardiovascular system, and there is some blood flow that maybe goes from the right to the left in this picture. And then maybe there is a target site over here, let's say a tumor, and we want to deliver some drugs to that tumor. Then the idea is that you might want to come in with a needle or a micro catheter or something to deliver some drugs to that tumor. But the problem is that the amount of pressure that you need to push a liquid through a small needle gets larger and larger with making these things smaller and smaller. And in fact, it scales to the 4th power. So that means that you make things 2 times smaller, you need 16 times more pressure. Then you want to make it again 2 times smaller, you need another 16 times more pressure. So, to the point that you need hundreds of, like amounts of forces. So it just becomes totally unviable. So instead of transporting things inside tubes, the idea is now to transport things on the outside of a filament. That is, you can build a bridge, and that bridge goes from your microcatheter towards your target site. And this bridge is now a very thin filament that's maybe 10 microns or so in width. And inside, you put these very small micromagnets. So all these little black lines over here are small micromagnetic particles. And then you've got your vesicles that will contain some drugs that will then be transported along this bridge, along this filament, using magnetic fields. So here is a video of exactly that process happening. You basically have a rotating magnetic field and therefore these particles will start rotating along the filament. And because of these micromagnets, they basically, let me go this way. They basically act as little anchoring points. So there is a very strong attraction force between one side of the particle and the micromagnet. And then as the field rotates, it will shift to the next stepping stone and the next one. And so it's a little bit, similar to these kinesin molecular motors that are walking along microtubules. Basically, biology has already solved this problem a long time ago. And so the question is, can we try to make something that biology has already invented, So now I have another question for you. Imagine that we try to do this faster and faster. So I'm going to rotate this magnetic field at different frequencies. On the top, very slow one, rotating at like 0.2 hertz. And then we try to, spin them faster and faster. And my question to you is, which one's going to move fastest? What do you guys think? Like somewhere at the top? The ones at the bottom? Somewhere in the middle? Yeah, okay. So what's happening? Here you can see them. It's a little Olympic race, if you wish. And so you were right. It's the ones in the middle that go fastest. Now, the reason why, at the top, they will certainly, move along slowly but surely. And, at the bottom, they will spin very fast, but they are slipping. So they can't really get traction with the surface. So you get lots of rotation, but no translation. And just, around the middle, around 1 or 2 hertz, you get the perfect, traction coefficients that will lead to the fastest possible motion. So you get some kind of maximum, right? If you plot, the speed of these particles moving along the filaments as a function of the frequency, initially they go faster and faster, then there is some kind of maximum, and then they slow down again. So if you're a doctor and you're trying to do some delivery for your patient, then you want to be in this area, right? And we can calculate exactly where that area is, because it's basically a balance of forces and a balance of torques. There's these, magnetic forces, and then you have also the viscosity of the liquid, and then you have the magnetic interactions with the surface. So if you balance those things with each other, it turns out that you can calculate exactly when that force balance breaks down, and that's when things start moving slower again. A way to look at that is the number of steps that you take for each cycle of the magnetic field. And so for the slow ones, it's always 2 steps per cycle. So basically, you're right, left, right, left, right, left. I was going to do, my shoelaces are in tights. I can't walk too much anymore. maybe I would have done a live demo for you, but I would have been in the regime on the right. So, you try to go faster and faster, and then you fall over your own legs like I always do. So yeah, that's what's happening. Okay, so the whole point of why we're doing this is because we want to see whether we can transport things against flows, right? Flow that maybe comes in from the right to the left, and we want to move the particles against the flow towards our target site. So we do that in the lab. We make these microfluidic devices. We put these artificial microtubules, these filaments, into the microchannels, and then we just pump a flow through there. And we see if we can drive particles against the flow. Now, here's a picture that shows you the behavior of these particles as you get larger and larger flow strengths. This is a constant magnetic field. And so, the particles are just anchored onto the filament. And now you see that if the flow gets stronger, they're kind of holding on. And then the flows get even stronger and they're holding on, on tight. And then these guys, they're like really about to detach, but they won't detach. They are very, very robust. They will not detach. It's just because locally these magnetic forces are so strong that yeah, it's all at the micro scale, right? So the scaling of these magnetic forces is like really favorable towards fluid flows. Here you can also, look at another plot, which basically shows how fast the cells are moving as a function of the flow strength. Here on the 0 line is the results that we had before. So that's just the speed in the absence of flow. And then here on the right is when they start moving against the flow. So they go slower and slower and slower. Eventually, they will move along with the flow, but they will not detach. So maybe in a cardiovascular system, you'll have some pulsatile flows, right? Like maybe the pressure is oscillating inside our vessels. But even if you go forward, you go backward a little bit, you go forward, you go backward a little bit, eventually you'll go forward and you will not detach. On the other side, here this is all moving along with the flow and there's no problem at all. They'll just move along. So again, if you're a doctor, what you want to make sure is that you are like operating somewhere in this triangle. So you don't want to go beyond this critical flow rate because then things cannot make it against the flow anymore. Okay, so we did some trials with this. These are not chemical drugs, but these are a physical type of treatment. It's a common cancer treatment drug. That's basically these very small iron oxide particles, magnetite particles. So the idea is that you bring these particles close to a tumor and then you put them into magnetic resonance. They start heating up and then you basically boil you kill the tumor by heat. So they're very small. They're about 1 micron in size now. So they're smaller than the actual filament, but they will walk together as clusters. So all of these little orange dots that you see down there are like clouds of these cancer treatment particles. And you can just transport them along the filaments using exactly the same mechanism. And here is another video of that. Yeah, here you can see like the particles are like, first they start assembling. There's a self-assembly process. And then once they get big enough, they start moving. They start rolling along the filaments. And so this is again, an example of collective motion. Individually, these cells or these particles are too small to move along the filament, but together they can. Together they can form these collective groups, and you see that the velocity increases as they form these groups. Here is another video, I think, of a little demonstration. So here we have some target site, and you can see the particles are walking along the filament, and then they get delivered, they get dropped off at the right location. And you can make this really small, right? So this is a microfluidic device. I think this filament is maybe, this one was a bit bigger, maybe 30 microns, I think, in diameter. So yeah, summary is basically that we want to compare this with other technologies that are out there. On this axis is like the size of the drug delivery instrument, and here is the speed at how fast they move. And so this one's very fast. This one's really, yeah, I think it's just because you get to move along a filament rather than having to swim through a liquid. So your traction is just better. So compared to these helical swimmers or other micro robots, the velocity is particularly good for these systems. The size is, maybe a bit large. So ideally, we want to move this point towards, the left-hand side of this figure. But, now we're on the order of, let's say, tens to hundreds of microns. And typical blood vessels are also on the order of, tens of microns. So if you want to go to, the micro capillary size, then that's kind of the size that we are aiming for. Maybe 10 times smaller would be ideal. And so I think that's basically all I have to say, except I have a little advertisement for you. Most of the things that I've been talking about is basically fluid mechanics, right? And so during the pandemic, yeah, like our lab closed. So I started doing some experiments in the kitchen instead. And I think the kitchen is just a great place to learn about fluid mechanics and to learn about science in general. So we wrote this review article to basically summarize different areas of physics using kitchen experiments. And so I don't know, here's an example of like a champagne glass. A normal glass will have these, turbulent flows that are like chaotically moving around. But you can treat these glasses with a little laser beam here, and then you get nucleation sites just at one location here at the bottom. And as a result, you get a very beautifully fixed flow pattern that will just stay there in place. And so you can see these stable vortex flows. Also, if you look at it from the top, you get these nice swirls moving around. And now you may think, well, that's nice and interesting to look at, but is it helpful? And the answer is, well, maybe, because, the champagne, these little bubbles, when they come to the interface, they pop, right? And when the bubbles pop, you get an aerosol. And then the smell, the aroma of the champagne comes to your nose because of that aerosolization. It turns out that if you stabilize these flow patterns, the aerosolization is increased as well. So the champagne smells much more aromatic in these, treated glasses. So yes, I would recommend this for a date night. And I don't know, there are these other things, like people have studied Rayleigh-Taylor instabilities, right? Where you have a heavy fluid that sits on top of a lighter fluid, and you get these plumes that sink down. You see that in cocktails as well, because alcohol will evaporate from the top. And so you get a layer of water, or, relatively more water, that's heavier than the alcohol, so you get these plumes as well. Now, Rayleigh-Taylor instabilities had been studied in detail before for, fluids that don't mix, like water and oil. But it had not really been studied in these miscible fluids. So there was this group of kitchen scientists that, they went all out and they built like a laser interferometer setup, with a beam splitter, and then, one beam goes through the sample, the other one comes around, you bring them back together again, you get this interference pattern. And from that interference pattern, you can exactly back calculate what the alcohol concentration must have been at any one location inside the glass. So this was kind of a breakthrough in terms of interferometry, and as a result, we now know how to study these miscible fluid Rayleigh-Taylor instabilities. So sometimes, science leads to better food, but sometimes food leads to better science as well. And maybe one more example. If you guys order a cappuccino here at the coffee shop, then the barista might make one of these beautiful patterns, right? Now, I'm a physicist, so I really don't know how to make these patterns. But I do know that there are a couple of variables that I can change. I can change the velocity of pouring the liquid, and I can change the height of my milk jug, and maybe the radius, of the spout. And so these are some numbers that I can put together into, dimensionless numbers. So the first one is the fruit number, which basically tells me about the ratio of gravity and inertia. And so you can think of these milk, patterns as inverted fountains. Normally a fountain is a heavy liquid that you push up into the air and then falls back down again. Here you have a lighter liquid that you push down into the coffee, and then it will rise back up again through buoyancy. So these inverted fountains you can study for different fruit numbers, for different levels of velocity and the height of your milk jug. And then that changes the color of your pattern, because the deeper you go, the more mixing you get. So the shallow one will give you really nice, like kind of whitish milk colors, and the ones on the right will give you these really beautiful, darker, brownish, coffee colors. So by varying that dimensionless number, you can change the colors of your pattern. Then there is, of course, also the Reynolds number, which will say something about turbulence or laminar flows. And then you get, of course, like nonlinear situations, right? So with a increasing Reynolds number, the fountain first gets deeper, but then you get so much turbulence that you get a lot of mixing. So yeah, that's it. So here is a table of contents, of this review article. We basically structured it like a menu. So it begins with, drinks and cocktails and starters, main courses, desserts, coffee, tea, and eventually washing the dishes. So with that, I say thank you very much, and thanks for listening.
Questions?
The effect of a particle on flow. So you did that with spherical particles. Extrapolate to bacteria. So when you add flagella, you change the shape of the particle. That impacts the progression of the fluid motion coming forward because it'll change the Darwin number in the back.
Yes. Yeah.
Or the fluid volume in the back.
Yeah.
Flagella.
Yeah.
Model that, or is there a way to model that in?
Yeah. That's a very good question. So in hydrodynamics, there is a theory that's called slender body theory that you can use to model, elongated objects. And so, yeah, that would change this drift volume. It will actually make it bigger, just because you have more surface area, I guess, or you can entrain even more fluid for rod-like bacteria.
Okay, so the flagella make it larger as well because they would sort of disperse, like in my brain, treating a very simple kind of person. I was thinking perfect, but they make that size bigger.
Yeah, that's right. Exactly. Yeah, like they add to the wake volume if you wish. Yeah.
Term where we're looking at the attraction of nutrients down to biofilms.
Yes.
Bacterial spreading. So does that take into account the capsule layer and that sort of like sticky glue that overlays?
Yes. Yeah, that's a very good question. No, so here we just take a single viscosity for the fluid. And it doesn't matter if it's like a really high viscosity, that wouldn't change the flows. But if you add elasticity to this fluid, that changes the memory timescales. And so that would, like that Ornstein-Uhlenbeck process I mentioned, would be slowed down. And so that means that you get more like slowly varying flows, which doesn't necessarily reduce the flux, but it does certainly change the timescales over which these flows will vary. And so I think that, well, in the end, the biofilm themselves, the bacteria are kind of embedded within this film, right? And it's really an elastic matrix. But it's more about the cells that are planktonic, that come out of this matrix and that start, looking for new areas to colonize. And they are not embedded in this matrix anymore. So they would be swimming in a liquid that is basically just water. And if they swim in the vicinity of that biofilm, then they would still stir everything up. So I think that's the better picture of looking at it. Yes.
Oh, for the nutrient delivery, with that dendritic structure that you showed, there were some of those dendrites that did not receive the particles. They might have been shielded by an upper branch or lower branch.
Yeah, that's possible.
They eventually die out, changing the shape, and then others get shielded, and ultimately with those dendrites, you're going to end up with the nutrient.
Yeah. So the idea is, I think that this is a very long-ranged effect, which means that you can bring in nutrients from really far, and after that you get local diffusion. So the picture that I showed you with the colors basically tells you about the active transport that's that is a long-range effect, and after that the ones that did not get so many nutrients from the active transport would still get it because of passive diffusion afterwards.
That race that you showed us with the 1 hertz and
yes
key frequencies. What I noticed is that the 2 hertz started out slower and ultimately finished the race faster than the 1 hertz, which started out faster. Is there any meaning in that, or that was just you know.
Oh, in the end, yeah, so a single particle will always fluctuate. So what matters is the average speed, I guess, and the distance, like, is quite a critical factor, because the magnetic forces, they will drop off very quickly. And so it might have been that they were just diffusing out a little bit, and then they came back in again. So I think, yeah, that was, and yeah, that's what your eyes have seen probably. Yeah. Yes.
In the first part,
I will also tie my shoelace.
cells are communicating with each other, right? By the contraction, right?
Yes.
By the
Yes. Yes. Ah, that's a very nice question. Yes. Yeah. Oh, yeah. So that's a really good question. So elongated particles or cells in flow tend to align with the shear plane of the flow, right? And so what that means is that the communication of the signal in one direction, you know, might be much more favorable than in another direction. And so I talked about percolation theory, where people typically study ordinary percolation, which means that all the directions are equally likely. But there is another universality class, which is called directed percolation, if there is some, you know, preferred directionality, especially if there is also an asymmetry in the shape of the organisms. And so, yeah, then you end up in a different universality class. Another interesting thing is that, you know, if you have two cells that are, let's say, arranged like this, right? Then if this one will contract first, it sends out a signal, and then it will, you know, communicate to this one over here. But if this one goes first, then the flow goes mostly in that direction, and then this one would not feel it. So you get, you know, arrangements where a signal can go from A to B, but not from B to A, which is basically a diode. And so if you have a network with diodes, you can make, you know, transistors, and you can make, you know, logic gates and everything. I don't think that these organisms will make the next supercomputer, but it's still interesting, right? Because these cells are a little bit like neurons. They're not, they're not multicellular in any way, but they are still communities of cells that are exchanging information, and in principle could also process information. And so I think that's fun to, you know, to try to understand as physicists, like, what are the limits, the physical limits of information processing in biology. And this is such a simple system that you could, you know, try to approach that from an analytical side as well.
Do we have any more questions? Okay, let's thank our speaker once again. Thank you.
Thanks.